Physiology of blood. Introduction. Functions of blood cell count and hematocrit, HCT, plasma proteins, blood cells, red blood cells, erythrocytes, white blood cells, leukocytes, platelets, thrombocytes. Blood is connective tissue. It is made up of cellular elements, also called formed elements, red blood cells, RBCs, white blood cells, WBCs, and cell fragments called platelets. Extracellular matrix, also called plasma, makes blood unique among connective tissues because it is fluid. Functions: deliver O2 and energy to the tissues, and remove CO2 and waste products. Defense and immune systems, regulation of temperature, transport of hormones and signaling molecules between tissues. Erythrocytes: male, 4.7 to 6.1; female, 4.2 to 5.4 times 1012 L. Packed cell volume, PCV, male, 0.41 to 0.52; female, 0.36 to 0.48 No unit hemoglobin, male, 130 to 180; female, 120 to 160 grams per liter. White blood cells, leukocytes, 4 to 11 times 109 per liter. Platelets, 150 to 400 times 109 per liter. Blood proteins, also termed plasma proteins, are proteins present in blood plasma. Total concentration of 65 to 83 grams per liter. Plasma proteins content, gram per liter. Typical functions: albumin 48, oncotic pressure, binds hormones and drugs, alpha globulins 5.5, copper transport, antiproteases, beta globulins, transferrin, prothrombin, plasminogen complement 3, 1, 0.7, 1.6, iron transport, hemostasis, immune system, gamma globulins 13, immune system, fibrinogen 3. Hemostasis primary structure is simply the sequence of amino acids in a polypeptide chain. Secondary structure refers to local folded structures that form within a polypeptide due to interactions between atoms of the backbone. Three-dimensional structure of a polypeptide is called its tertiary structure. The tertiary structure is primarily due to interactions between the R groups of the amino acids that make up the protein. The normal healthy adult produces 119 million red blood cells per second. It forms 44% of the total blood volume, and a single RBC cell is sized 0.00007 meters. They are produced by red bone marrow via a process called erythropoiesis. Lifespan: 100 to 120 days. The shape and flexibility of red cells allows them to deform easily and pass through capillaries. Importantly, they contain hemoglobin, which is responsible for carriage of O2, and also plays a role in acid-base buffering. WBCs have a nucleus and lack hemoglobin. WBCs form 1% of the total blood volume in healthy adults. They help fight infection and defend the body against other foreign materials. Function as a cleanup crew that removes worn-out cells and tissue debris. Neutrophil, neutral staining granules, innate immune system, major component of pus. Neutrophils live for 6H in blood, longer in tissues. 50 to 70% total, 3000-6000 by 106 per liter. Eosinophil, acid staining granules, less motile but longer lived, increased in allergic disease, releasing inflammatory mediators, e.g. bradykinin, serotonin, etc. 1 to 4% total, 150-300 by 106 per liter. Basophil, basic staining granules, release histamine and heparin as part of the inflammatory response and are similar to tissue mast cells. Approximately 0.5% total, O-100 by 106 per liter. Agranulocytes, mononuclear, lymphocytes originate in the bone marrow but mature in the lymph nodes, thymus, and spleen. B cells produce gamma globulins. T cells coordinate in immune response, and K cells kills infected or cancerous cells. 20 to 40% total, 1500-3000 by 106 per liter. Monocytes, phagocytes, but larger and longer lived. After formation in the marrow, they circulate in the blood for 72H before entering tissues to become macrophages. 2 to 8% total, 300-600 by 106 per liter. Platelets are the cells that circulate within our blood and bind together when they recognize damaged blood vessels. 1/4 of RBCs, 2 to 4 micrometers in diameter, irregular shape. Blood clotting process. Hemostasis is a way of the body to stop injured blood vessels from bleeding. Hemostasis has three major processes, namely the constriction of blood vessels, activity of the platelets, and activity of the proteins found in blood, clotting factors. Needed for clotting. Clotting factors are components found in plasma that are linked to the blood clotting process. The clotting factors are factor I, fibrinogen, factor II, prothrombin, factor III, tissue thromboplastin or tissue factor, factor IV, ionized calcium, factor V, labile factor or proaccelerin, factor VII, stable factor or proconvertin, and factor VIII, antihemophilic factor. Additionally, the coagulation factors also include factor IX, plasma thromboplastin component or the Christmas factor, factor X, Stewart-Prower factor, factor XI, plasma thromboplastin antecedent, factor XII, Hagerman factor, and factor XIII, fibrin stabilizing factor.